Let's get started. It's my pleasure, great pleasure, to introduce Dr. Bruno Andrade here from Brazil. Bruno currently is, if I can pronounce all this correctly.
It's in the slide.
Can read it, but anyway. He's a full professor of pathology at the School of Medicine, Faculty of Technology at Ciência.
Yeah.
Okay. At Salvador in Brazil. He is currently the PI of Report Brazil, and also plays a very important role in TB-RIC, where we have this meeting today and tomorrow. Bruno obtained his MD degree from University of Bahia, and then his PhD from Cruz. He then moved on to the NIH and did a postdoc in human immunology with Alan Scherk. He spent a few years there and then went back to Brazil, and that's really, in a very short time, has become a star in the field of TB. He's really achieved a lot in a very short span of time, very early in his career. He has a number of NIH grants. It's a long list. He has lots of funding from various funding agencies in Brazil. Recently, he's also become Dean of Research for an initiative with the UAE and Brazil, where they're setting up large numbers of medical schools all over Brazil and building infrastructure for research at all these schools. So he's really heavily involved in getting this whole program started in Brazil. I'm not sure how he finds time for all these activities. He also has a very large group of very enthusiastic, smart people who really carry his vision of research. He has large numbers of publications, 250, most of them in very high-profile journals. So clearly, it's a privilege to have him here talk to us on his ongoing work at his lab. Welcome. I was trying to find out the word in Portuguese for welcome.
Bem-vindo. Let me see if I can make this work. Do I need this because the microphone is here?
Maybe,
Yeah.
Can you say so? Can you guys hear us, people on Zoom?
Yes, we can.
No, it's fine. Let me try it without this. It's a great pleasure to be here. I'm a little bit tired from the trip. Several trips. I've always been willing to come here to talk to you guys and discuss possibilities. And that's why when Padmini invited me to come a little bit earlier than our meeting, I was happy. This is not a talk about TB biomarkers, okay? I needed the title, so I sent this up. But the idea is to discuss very informally, and you guys can ask whenever you feel, to show what we are doing right now in Brazil and in other places. So, and we have the king of biomarkers sitting right here, Oswaldo. So, just as a disclosure, I have several appointments now in Brazil, but three main appointments that provided data that will be shown today. I have a lab at Cruz, is the Brazilian NIH, the part of research of the Ministry of Health. Then we have the consulting company that became recently an NGO, and this company was bought 80% by the Mubadala Capital, which is one of the largest investment groups in the world. And what we do here is we create a program to develop careers of young physician scientists in the country, and we expanded for the entire South America, and now we're expanding to South Asia. And then, as Padmini indicated, I have this appointment with the holding company that has several medical schools across Brazil, and the idea is to create this network of schools linked towards medical research. How we divided the jobs and how could we make that happen? We And Cruz is vastly, dramatically involved with the translational science work that we developed. We have several platforms, single cell platforms, cellular immunology platforms, and proteomics. And that it's very widely used in several projects, including those related to Report International. We have the analytical core that has a lot of young investigators, roughly 120 people working into developing scripts and ways to show and digest complex data. I will discuss a little bit this part of the work. And then in the Clariant education, we have several clinical research sites across the country, and that's where we recruit patients from a wide range of infectious diseases and non-infectious diseases as well. And I could join those appointments through collaborative agreements. I can hear my voice, so someone has a microphone open in the Zoom. So the scope of work, what we really do, we try to understand susceptibility to disease. So one example here, if a person is exposed to TB, some of them do not develop anything, right? Even infection, the mycobacteria cannot establish in this person. So we call resistance to infection, it's fully resistant. And maybe it's affected by genetic factors, but it's vastly unclear all the mechanisms involved in this phenomenon. So among those that are susceptible to infection, then we have also several possible manifestations from resistance to disease. It's a person that is infected and has no symptoms or even damage, no damage, and also those who have different degrees of susceptibility. So that intrigued me first in malaria. When I started my career, I was really into understanding disease tolerance in malaria. Then we migrated to TB and we realized that it's far more complicated. This is a recent review from our group together with you guys from Rutgers here. And the idea is that, well, we don't really understand the clinical spectrum of disease. Every year, a new manifestation is described, and we are trying to understand the dynamics between the bug fitness, the damage, tissue damage, and how we control damage, and how we manifest disease. And the bottom, we just show the lack of useful or clinically applied and approved biomarkers for TB disease. So we have the expert here being released or approved in 2010, and then a huge gap in time, and then many other things arriving. But there is a lack of biomarkers that are validated and used to the level that we need to fight the disease. Using the expert, for example, we recently published a paper. It's a small paper in Report Brazil network, but demonstrating or suggesting that the cyclothreshold value in a person that has a positive test for TB could infer risk of transmission. And so contacts from people that have very low cyclothreshold values, suggesting a high bacterial burden, they more often converted from IgRA positive or negative to positive, suggesting some degree of transmissibility. It's just an insight of how to work on previously published biomarkers or tools or platforms to come and extract more information. So the TB biomarkers, I show this slide quite often, has dramatically changed the way that we approach the biomarker research from the identification of the bug in the history to recent several launches of new technologies and platforms that investigate several aspects of the immune response, mostly immune response from the host side and fewer from the bug side. And now we have generation of very complex and large amount of data that needs to be sometimes deconvoluted, merged, cleaned, and interpret to infer something or a biological process. And that's where we are actually expanding and publishing a lot of papers, but not really progressing towards implementation of a useful biomarker. And that's what I think report can actually help. If we have a very well-defined clinical platform, we can test those technologies and see what's good or not. And that's why Shito and Padmini are leading such a program. If we look at science, and I struggled a lot being a Brazilian trying to study TB after a time in US, there was no money. And what I found in Brazil, and when I look at the US, actually you guys are kind of similar, we have silos, several different silos. The statisticians in one side, basic scientists and assistant physicians, clinical researchers, medical engineering departments, they're all excellent in what they do, but they don't really talk to each other. And a lack of communication is there. And because of that, we can't, or it takes a long time to translate basic knowledge into product and application because these silos are too busy thinking about themselves and how to survive in the funding mechanisms that the research demands. So this is a key problem for me, and we're trying in Brazil, because there are very few people interested in this topic, we were able to merge and to create a platform that can promote discussions across these silos more efficiently. And I will show a couple of examples. So the other problem is that we don't understand TB, right? It's a pretty old disease, one of the oldest identified in humanity, and we don't really know how it works and how damage is caused and what are the factors driving susceptibility. We have no idea. And when we started looking at the evolution of the biomarker studies, what we see is that, well, from the minimalist point of view, when we pick up a molecule and test at different clinical groups to see if that level is up and down, and we can exclude some confounding factors mathematically, it's not super ideal, but that's what we have. From that point to some more complicated analysis with adjustments and trying to reduce the noise. And then the multi-omic platforms came to the field showing that things are more complicated. So it became harder to clean data and to understand what is really a biomarker doing or how a molecule is participating in the process. So one example is interferon gamma that became a bad guy recently. People are bad-mouthing interferon gamma, like maybe it's promoting disease instead of controlling disease. So that just demonstrates that we know pretty much nothing about disease pathogenesis and how to come up with a product that can help clinical decision if we don't understand the disease. So instead of trying to find a key molecule that could serve or a signature that could serve to identify patients at risk, maybe we can dedicate more time and more funding to studies to study fundamental questions about pathogenesis that's very lacking. And the disease models don't really contribute much quite often to understand of human disease. So things are just complicated. And the other point that we talked when a young investigator wants to start to understand biology in TB is that, well, biology is what it is and not what we want it to be, right? So many from the biomarkers field, they really face that as their favorite molecule or favorite signature, and they just want it to be validated. And the truth is, we should not look at the data this way, right? Let the data tell the story. And it seems so obvious that, but it's so hard to discuss with people about these topics. And the other thing is to recognize that many factors that are not measured are influencing our results, right? There are many ways to test those interactions. But the thing is, if we don't really, we can only extrapolate to a certain point. And many factors influencing disease signals are being considered ad hoc after the signatures were published. And a simple factor is sexual hormones, how testosterone or estrogen could mess up these expression levels that would explain partially or not. And just a simple exclusion or weighting those factors mathematically might not be sufficient to actually have a very clear-cut definition of what the influence is. My group works with different tools to visualize data, just like many others, including Evans, that is sitting right here. Very good to see you here, man. And we try to see how these factors connect directly or indirectly using different tools. Bayesian network is one of them, but it is still, it's whatever we measure. And, we might not be measuring the right thing or considering the right factors because we don't understand disease. Anyways, so to try to zoom out, instead of being really dedicated to find the best molecule, the best factor discriminating patient groups, we wanted to zoom out and say, like, how these things connect to each other and how they relate. And I don't really care what the outcome is. I just want to understand. And the work that we developed was compared by many groups, and this was a recent interview with the pointillism, which is if you zoom out, you kind of can see things, but if you zoom in, it gets really messy and we don't really understand what's going on. It's pretty much what's going on when we combine all the studies about TB. Couple of examples here. We initiated trying to understand interactions between tuberculosis and diabetes. We measured a couple of markers depending on availability in commercial kits. And we had no formal hypothesis by there. And we measured everything. We were just measuring and comparing groups and seeing what's going on. And when we pull everything together, we could discriminate the groups. The groups are fairly different when we pull data together. So now what we do with that, it's complicated to create a signature and it's probably full of noise. So if you play with the system, put in more variables, removing variables, you could test the influence of combinations of variables in trying to explain this. This was just the first try to get that prior to establishment of the field of multi-omics, okay? Then using now multi-omics, we got better in managing data and trying to discriminate groups. And although the statistical tools and data visualization tools were different and kind of evolved, we still fail to understand what is really driving differences between the groups. So we measure proteomics, transcriptomics, and we fail to distinguish TB diabetes from TB non-diabetes in this particular group. We could distinguish TB from non-TB, but we don't really need multi-omic measurements to tell who is infected or not, right? So we could look at other things that people fairly neglect, like non-coding RNA. And well, it results in fancy studies and colorful figures, but what's the information that we can extract that can be useful? So this is what is lacking. I think the field dramatically needs more thought and more discussions about what to do with the biomarker data and how to explore. And in this journey, what I figure is, well, it's if you measure things and you calculate things and you plot graphs, you will find a lot of things, and then they are not validated because the sample size or the patient evaluation changes dramatically. Some people from this field, they actually agreed, or their view is completely different than mine. They think that we need noisy data to claim if something shows up in noisy data, it might be useful. People from Stanford, they published a lot of papers suggesting that if you take data from studies and you really don't care for how the patients were evaluated or the things were measured, and things resist to noise, this may be more informative than well-described studies. Look, I tried, but what I realized is if we don't know what kind of patients you're studying, the information that you extract from that, it's not useful. So I'm actually in the opposite. We need conglomerates of medical and clinical research sites very well standardized, so we know exactly what we are measuring and from whom. But again, zooming out, another way to look at those measurements is to stop trying to distinguish clinical conditions, but trying to understand biology and how these molecules talk to each other. So when we first plot these graphs, an investigator from NIH and also from Hopkins, Tom Quinn, came to Alan Scher's table and said, like, this seems a ballistic exercise from the technical police, right? So what these lines represent is how a concentration of a given marker relates to another one. And when we zoom out and look at the overall profile of these relationships, we could see that different clinical presentations could lead to different profiles of communications. So if you look at if you consider the immune system as a homeostatic system that needs to reestablish homeostasis upon a disturbance in the system, it's easy to think that things needs to be coordinated. So if you're producing one cytokine, that will influence positively or negatively the production of other things, and the signals are shut down, other signals are stimulated, and that results in matrices of correlation that are now used to understand pathways. So which pathways are activated or deactivated? That is measured by quantifying the profile of correlations between expression of molecules. And we started this in kind of limited proteomics or Luminex assays a while ago. We expanded these studies to show that the profile of correlations could actually tell us about treatment stages. As you can see here, I have 3 different networks from the same participants from a cohort with Robert Wilkinson from South Africa. And what we found is the system is really shut down, meaning that the number of correlations really reduce after TB is treated. So something is going on, even during treatment, that keeps the immune system active in a way that we can measure or quantify or qualify. We can compare number of correlations, strength of correlations, and all that. And we could use that information on how the network behaves, and we can calculate this per individual and actually predict outcomes in a separate cohort that is listed here. This knowledge about how molecules interact, and instead of looking at one single molecule or a combination of molecules, we look at everything, and we are not interested in concentration data or expression level. We are interested in how they relate to other concentrations. And when we ignore the concentration data and goes to correlation data, we could really gain more information about pathogenesis. So one example here in monkey granulomas is that, well, we find that when we inhibit PD-1 in monkeys infected with TB, then there is a disturbance in the network or how their immune system is behaving that can be read or measured by quantifying those correlations. And we can then rank the importance of such markers and come up with a combination of them that are driving the complexity of the network, meaning how the immune system is communicating.
When I look at the isotype and PD-1,
Yeah.
The players are the same. Right?
Yeah, exactly. But that was on purpose. We wanted to show the same players, but how they are communicating to each other, how they are relating to each other.
Oh, you see more lines.
Yeah, and the studies from our group, when we do this in several different cohorts and disease models, what we find, Padmini, is that when we have a lot of correlations going on, that means that there is an activation of the immune system or perturbance of the immune system, then things tend to correlate to shut down or induce more disease. The sparse canonical correlation analysis, after we used the first time, I will actually show the next slide, the paper that used biomarker data using that approach takes into account how these markers correlate with each other rather than their concentration to explain clinical profiles. And here you can see that when you use correlation data, just like a PCA, but as in a different mathematical calculation, you could distinguish the groups, and we can rank the importance of each marker in this network that can explain the differences. This is the first time we tried. It was a Nature paper with Catherine Meyer-Barber from NIH. And we took a lot of different markers and we put them together. And instead of asking which markers were up and down, and was like how the relationship between these markers or concentrations between these markers could explain different TB profiles, right? Severe disease that in this particular study we defined as positive smear and bilateral disease. I'm just trying to show how to you guys how useful other approaches rather than just measuring or quantifying concentrations of biomarkers could give us tips of what's going on.
So when you say correlation, it's based on what? If it's not based on that they both up or
No. The concentrations is used, but I don't care. I don't consider the concentration itself to predict the outcomes. I use the correlation data to predict the outcomes, right? People from the field use a lot of PCAs. PCAs, as everyone knows well, it's not really designed to discriminate groups. It's designed to explain variance of the data and differences in variance of the data. The canonical correlation analysis puts on top of that the idea of discrimination. So some softwares or some scripts will use that as a discriminant analysis, right? So, but what is imputed in the data is the correlation between the concentrations rather than the concentrations themselves. Aside from the health field, this approach has been used for a while to predict the weather, right? So when I was a kid, my dad, when I was 15 years old, my dad gave me my gift. My birthday gift was money, which was good, but I had to use that money to in the stock market with my dad. And he wanted me to use statistical methods to prove to them which stock I would sell or buy. And my brother and I used very different approaches. My brother invested in that time machine learning, and he was more successful than I to predict the stock market. And I wanted to learn mathematics, so I used weather prediction analysis that was based mostly in forecasting, but also in canonical correlation. If you want to tell the probability of rain or snow tomorrow, if I give you data on temperature alone, doesn't tell you anything. Humidity, it doesn't really say anything. But if I tell you temperature, humidity, data from previous years and put this together in a correlation matrix, that generates the probability. We still fail, but it's just a tool to say. See, but how different it is from a basic up and down expression level to see how effective can we distinguish. Sometimes you don't gain power to predict or to translate into a clinically useful biomarker, but sometimes that gives you more of an idea of what's happening in terms of pathogenesis, at least in my opinion, okay? That's why I decided to show to you. Interestingly, I decided to use the canonical correlation in this field. Until that time, until 2014, my knowledge about statistics were not really linked to health. I was using that to make money, not to be a scientist. And when I showed to my dad a PCA plot to show those discriminations, he said, like, look, my dad is in the acknowledgements of this paper because he was like, you're not using the right analysis to actually make a point that you want. You need to use a sparse canonical correlation. And I said, huh. And then after that, I consult with him without paying anything until today. But the idea is to come up with different statistical approaches with the same data that we are used to see what kind of insights can we gain when we do that. And it's more of an exercise for me and my team now. Strangely enough, after we published a couple of those papers with crazy, colorful figures out of crappy data, many universities around the world, but also UN and WHO, they actually started having us as contractors to digest their own data and tell what investigators at Hopkins, for example, could do with their data. And we use that money to support the career development program that we have. And then we hire more crazy data scientists to feed more into that. And it's now a complete self-sufficient structure that does not depend on grants to support careers. When we published this paper, we got this, I think, intrigued a bunch of people, and we got even an editorial in the New England and other Nature journals suggesting that, well, maybe we should look at the biomarker data using those tools to see if we can gain more insights, not into prediction power, but into understanding disease. And that leads to a patent, analytical patent, but also an intervention patent. This is an interesting study, not a high-profile paper, but we took a large cohort that was performed nested to a clinical trial that was performed in Mexico and South Africa using CCR5 inhibitor, Maraviroc, in patients with TB and HIV. And what we found is, and if we pre-treatment, at baseline, if we measured biomarker, and it was a lot of Luminex assays performed in a single lab, in a centralized lab from samples from these two countries, we could tell who was from South Africa or Mexico based on the expression profiles, right? How these biomarkers correlate, suggesting that, well, maybe there is something about living in at least these two countries that is changing the way that these molecules relate to each other. And it's something that if we just measure the concentrations and we don't look at the way that they relate, they seem very similar. But we actually can tell with a very high degree of accuracy, a sensitivity of 90%, specificity of 86% to tell who was from Mexico or South Africa. So this is all interesting and at least graphically appealing things, but it's not all what it's about, right? In order to survive in this field in Brazil in 2015, when I went back, there was no money available. There is nothing, no infrastructure that needed to be, and everything needed to be built from scratch to have a solid program on TB biomarkers or even data analysis in TB. Brazil is a large country, has a lot of very competent people, but very few genuinely interested in TB. If we just correlate with the size of the country. So what I tried to do, and I'm still in this process, I'm still trying, is to establish networks to isolate pathogens and understand the biology of TB in Brazil and disease epidemiology and determinants. So from scratch, which regions in the country deserve that? And I was lucky enough that Report Brazil had been implemented 2 years earlier. And I found my way to join the network and to contribute more. My first role was just to help the labs to be established. And then with time, I became the PI, and we build more and more research infrastructure in the country. But aside from Report, we have our own other research sites that are mostly hospital-based sites that are contributing with research activities as well. And not only TB, but in a wide range of infectious diseases. That costs a lot of money. And that's why the need of having aid from the Mubadala capital was so important. So the current budget that I have for investments in research infrastructure is $10 million a year for the next 5 years to build things and integrate them and harmonize protocols. And again, Report was very important because I jumped several steps trying to harmonize things not Report related due to the experience that I gained or we gained with Report. The other thing that we decided to do is Brazil has this advantage to have a national disease registry databases very large databases and well documented. Well, as all or any registry database, it has problems, right? But still, a lot of with a very large number of diseases, all the infectious disease that requires notification and to get the treatment, TB is one of them. They have that, and we built a data center that downloaded all the data, and we develop a recursive system that every time a new notification is performed, our system downloads that and integrates and cleans with what we already have. So we have ongoing feed of that data. And I'll show a couple of things. During COVID, we helped the government to build what we call situation rooms, which is to design dashboards using this national registry programs that a policymaker could see changes in trends in disease and treatments and testing in real time. And that was critical for COVID decision-making strategies, but then we expanded to a variety of other diseases. And this is very would keep the groups monetarily as well, because that we charge as a contractor. And with those initiatives all together, we were able to concatenate, to merge funding from different agencies to support the group. But what I'm really proud is not about the data or about the programs, it's about the people that we could help. And many of these are all physicians now, but they were medical students at that time. And just the top students that I had in terms of publications and grants. But we have more than 100 now that are becoming more successful. And many of you guys know already some of them that participate in meetings like Beatriz. And they become independent. And my job becomes now how to support them, which is a complete different game. Beatriz decided to study indigenous populations, and she got a 5 million grant to study different populations in Brazil, integrate for TB screening and TB biomarkers. And that just showed to us how complicated it is to start a new research line with a complete different populations from scratch. So we're still learning, but it's been worth it. We build partnerships with a lot of universities to integrate with our network of universities, and that allowed exchange of professors, students, and this is something that has helped the group to gain more insights and maturity to keep the projects going. Some ideas are just crazy, but as I said, exercise, and we started running with data that we already had. One of them is this molecular degree of perturbation, which is inspired by a metric called molecular degree from health, which measures basically in overall quantification of disturbances in concentrations of markers that will altogether show how distant you are from a reference group, okay? So in this case here, I have people without TB and without diabetes. We establish as a referral group or reference group, and then we can, I'm not going to describe all the details, but you can tell how distant this person is with an advantage from the old metric that is to take into account. So I can use the MDP values from one study and compare to other studies due to the matter of how these things were calculated, whether the molecular, the distance to health actually needs to be calculated for each study. So all I'm trying to say is that researching or trying to digest data from biomarker studies led us to come up with new approaches, at least for us, that will facilitate comparisons between the studies and how we can understand now disease pathogenesis or disease severity or clinical presentation across the studies using one single metrics. We could put MDP in a correlation matrix and now measure or assess which markers are driving this metric more or less. We kept our hands in basic science as well, and together with folks, with Ellen, Alan was very generous with us to allow us to keep collaborating and publishing with him. And several interesting papers came out. And our participation, aside from designing the experiments with them, was actually to digest human data from tissue, from different sample sets in different cohorts to make a point about the involvement of the molecules that they were interested in disease pathogenesis, and including single cells and other things. Such studies led us to develop spatial transcriptomic analysis in a different way to come up with graphical manifestations. This is a study that we use AI-based spatial omics to define regions in the tissues or granulomas that are being important for SIV, TB co-infection. That could go on, and we can just show, we can deconvolute the data from spatial transcriptomics and show associations of molecules, run those crazy correlation graphs and show how this is different or differently modulated by the clinical condition. It's just to show that the exercise of using machine learning-based tools to spatial transcriptomics could change the way that we look at the spatial transcriptomics or single-cell omics as well. Using machine learning algorithms could lead us to also identify markers that could predict things and propose markers. In one study, we used anemia, like low levels of hemoglobin, as a marker, and we show how these things could be used to predict TB onset and iris and death in different cohorts. And also with other collaborators from South Africa, we show models that could predict early death in patients that are hospitalized with TB HIV. But more clinically oriented data science using information from Report Brazil and Report International. This is Report Brazil. We have 5 sites. We reduced for 4 sites now. And those are the people involved. I won't have time to discuss all that. Those are just examples of funding that we got, totalizing a couple of subcontracts with Cruz, with a lot of money to study different aspects of TB pathophysiology. Those are not just money from report networks, but all the studies that involve the sites that are included in report. This is the collaboration network. And using simple data from EPI forms and with REDCap, we could publish a couple of papers, playing with prediction models to identify patients at risk of failing treatment. We could predict the type of outcome, subdividing them by type of bad outcome. We could test prediction models for adverse drug reactions and other problems during treatment, just using EPI data. I think this is very interesting. We could identify patients that were at risk of not completing the TB cascade of care in context, and which are the characteristics that will lead these people to fail or not being treated or to just be lost to follow-up. And by identifying those, we can inform the government. And as expected, this type of publications were more useful for the Brazilian government than the biomarker publications. We've done this in children as well, to show which characteristics will lead to children not complete the cascade of care. And more recently, we kind of compared our cohort to the Brazilian National Registry, and we found a lot of similarities. And then by linking the data, we could track our patients and see what happened with them, even though they were lost to follow-up in our cohort. So we can see if this person developed TB elsewhere or was in a different site or city, if it had kids, if it received the government incentive programs, and we could have access to anything. The data center is part of the Ministry of Health. So we link the data for the Ministry of Health. We de-identify, and then we perform our analysis and And with that, we were able to run a lot of projects that we were underpowered with Brazil. So original ideas from Report Brazil were tested in this large 1 million plus people, and we found characteristics that were associated with isoniazid monoresistance, and how isoniazid monoresistance could actually lead to unfavorable outcome. And we found that this was the truth. It's mostly driven by death. The data linkage costed us to now estimate a cost of TB in Brazil in a resolution that the government did not have. Now we know exactly how much it costs to treat one patient with pulmonary TB. And not only that, if we break down by type of vulnerability, we could identify the subpopulations that have the highest risk or for failure. But not only that, how much should you invest in different types of vulnerable populations to achieve the goals defined by the WHO? Everything with data linkage and machine learning. So we found which are the most vulnerable populations. And to our not surprisingly, we found the homelessness is the top vulnerability associated with unfavorable outcomes. Then we go down and describe exactly what is within this population is driving the susceptibility. They're all published papers. We use recently machine learning algorithms to understand what is driving loss to follow-up. So this was just one exercise comparing different machine learning tools to identify characteristics that will inform risk of loss to follow-up in patients in the entire Brazil. And it was not fantastic, but we could actually tell who is going to be lost to follow-up with overall 70% of accuracy, just using basic data from the registry databases. Finalizing. So everything was built, as Padmini pointed out, in a very fast pace, right? So we started our business in Report Brazil in 2013, and we have now the largest TB cohort in the country, almost 2,000 people, and with close contacts as well. We have a huge biorepository with all kinds of samples, and is available for collaboration, of course. And then the phase 2 that just got approved the end of 2022, funding started 23. We expanded those aims, but we added something that I believe that was worth, and I'm proud of that. That is a program to develop the next generation of TB scientists in Brazil. And that inspired Report International to implement its own program. It's called a REACT program, and we select 2 fellows, 2 postdocs, and 2 young scientists with funding, mentorship, grantsmanship course, scientific writing course to try to push these guys to a different level. And we've been successfully doing that. Aside from precision ID, we have many other venues that I just added this, Padmini. So if someone here is interested in these topics, we could discuss. My wife is a clinical dietitian, and she works with autism, and she is actually the head of the institute, not me. And she analyzes diet profiles using correlations rather than grams or total grams that you eat, and how your diet correlates. And a bunch of interesting studies came out just showing that, well, trophic people, when you eat more of one given food group, you end up eating more of another food group, and the correlations are positive. And when you see disturbances in this profile, meaning that you will have selection of food intake, you eat more of a group and less of another group, that would define overweight children or adolescents, suggesting that the diet behavior is being highly correlated with disturbances in weight. Just an analytical exercise, but we call that systems neutrology, that is using systems biology and big data tools to understand nutrition. Arthur and other people from the group are pretty well-trained in immune metabolomics, and we use that more and more to define disease states. I will show an interesting graph with an example on how to visualize disease states, systems immunology in sickle cell anemia, systems biology in children with autism, but also in elderly with Alzheimer's and other things, single cell multi-omics, not transcriptomics, but multi-omics in grafts versus host disease, immunogenetics and of antimicrobial resistance, and then the interplay between diet, microbiome, inflammation, and gestational outcomes. Everything is possible because our group is now close to 300 people. So we could study those other things. Let's see if I can. So this is just one example of how we could, oops, it's not going. Anyways, I can show to whoever is interested later, but it's basically a three-dimensional field that we can identify patients with different disease by multi-omic profiling, and we can show how different diseases are associated with different clusters of patients. It's just to, but it's a very interesting tool that we are incorporating now spatial transcriptomics into that. So then what we believe that we are doing in Brazil, at least, is a way to make those different silos to communicate. And we believe we are achieving that because we have metrics to show that, well, we have a huge multidisciplinary team with diverse expertise and complementary skills, developing data visualization tools dedicated to get better at this and continue training of physician scientists. When we were being interviewed by a local journal, they were like, why physicians? Is that like, why, what do you have against the other specialties? And I was like, I don't have anything against that. We do have other specialties. Mariana is not a physician, but there is no physician scientist in TB in Brazil, very few. So that's why we need to invest more at this, and we've been able to do that. And capacity to links layer of complex data in a country that we're not doing this traditionally, and to digest and inform policy-making strategy. So in the next RIC meeting, we will have a heavy presence of the Ministry of Health, mostly driven by the idea that report helps the government to take better decisions. And we've been able to show this. Going back to biomarkers, I will just end with this view that we have, and we concatenate it into this table that were published recently. What are the major challenges in the field in our opinion? Well, we have different people in different countries, and I showed to you guys how biomarker profiles or relationships between them are different between folks from South Africa or Mexico. Well, we need large sample size, geographically diverse populations, but harmonized protocols, multinational collaborations, and precision medicine approach. Well, having that in a single package is hard. We know how hard it is to get funding, and each silo, again, is thinking about their own survival mechanisms. So it's hard to put people together. And I think Jerry and Rick, together with Tim, Amita, Padmini, been successfully doing that in such complicated setting, which is Report International. We need international consortium, in my opinion, and we need to find a way to communicate with others. That's for you, Jerry, how we can expand our consortia with other groups. Centralized labs, Rick was able to implement that view successfully. We are going to test in the biomarker study. Standardizing, sharing data policies, and Stephanie is it's critical in this field. But as you see here, everything that we listed in this table has to do with the idea that report can actually make this change. So this is not my group. This is what we believe that we can do and showing directions. thank you very much. Those are the fundings that supported all the studies. And this is just a very few of the key people that I presented data from. Arthur, many of you guys know, but he is a senior scientist from the group leading the data visualization platform. Beatriz, it's a physician scientist, young physician scientist with indigenous populations. Maria is doing a postdoc now at Vanderbilt. Mariana is the program manager from Report Brazil in systems biology from the group. And we could not fit everybody in a single picture, but I acknowledge all of them here. Thank you very much, and I'm open for questions.
Anyone from the Zoom calls? Any questions? Evan.
So Bruno, great talk. Thank you. Thank you for sharing all that big picture. That's fantastic and amazing work. So if you go back to your challenges, I'm definitely biased, right? Because you've got these potential solutions. What data problems and data solutions are needed as well, right? I mean, you're going to be, this is how do we get good data?
Yeah.
Right. But then what are the challenges after that?
It's a pretty good question. Well, first we will know when we have data. I think the challenges are how, although the clinical protocols are harmonized and the measures are performed in a single lab, the challenges in handling such a large multi-platform data and how to clean the data, and at the end of the cleaning process, what will be the quality of the data? We just don't know, right? I think the RIC biomarker protocol will likely be the largest multinational study to perform such simultaneously measurements of everything in terms of numbers. But so I think I guess we will see what we get. But just an example, although we have everything standardized, some labs have extreme difficulty to produce good information, sample related, or sometimes just the teams. And that is explained by another layer of complexity that I haven't explored here, which is how do you support the research teams, right? How do you have a stable team? So what happens is if you keep changing the lab technicians all the time, although there is an SOP and a constant training, we face challenges. So I received a package from samples. I'm not saying the site from one of the report sites, but the cells are all dead. There was nothing to do. And can you imagine the money that was spent to do all that? And then you cannot do anything with the data. So I think the challenge is, Evan, we will need you, right, to discuss all that. But I think we'll be mostly with how to get a digestible database integrating all these platforms that we are measuring in a way that is able to inform things. But it will be fun to do it, right? Yeah.
Hi, I have a question. Hi, Bruno. This is Maria Gennaro. Can I ask now, Padmini?
Hi, Bruno.
Great talk, really thought-provoking and stimulating, and wonderful progress both in the content and really great talk. I really enjoyed it. I have two questions. One is related to what you just said. At some point during the talk, you said that there are other investigators that kind of try to or purposely disregard noise because that's what the data come with. And I think I know who you're referring to, but regardless of who it is, but you just now you mentioned the fact that there are challenges even when you set up and you have the resources to set up laboratories that in principle are well harmonized with each other, follow the same protocols, etc., etc. There is noise there too. So in the real world, there is even more noise. So, I mean, isn't noise part of, is not uneliminable? That's the first question. The second question that I have is, you also mentioned that, you also said at some point that we don't know enough about TB pathogenesis. And, you know, I agree with you, but at the same time, you know, we've a lot of people all over the world, they've been studying TB pathogenesis for, you know, for a while now, with utilizing a very considerable amount of resources. So if there are still things that we don't know, the approach that you're proposing couldn't in fact be the way to actually discover pathogenesis rather than having to rely on pathogenesis. Because if you have, when you find these correlations of markers, they could be, you know, immune response markers and metabolic, you know, whatever it, you know, the how can I say, the multi-omics, or, you know, the multiple classes of markers that you use, couldn't those be used to discover pathogenesis rather than waiting for the pathogenesis studies to tell you what markers to look at?
Great questions. Thank you very much. The first about the noise, I don't think we can eliminate noise. Noise is part of the business, and the thing is the amount of noise. if you look at the super noisy databases, let's say like that, it will be even harder to delineate associations. That's what I'm trying to say. Look, Report has invested a lot, but we do not have we will be discussing that tomorrow, but we don't have a mechanism to support financially the teams in a way to make sure that everybody's doing what they're supposed to do. So that is a noise that is pretty hard to account for, but we need to consider them. I think one of the challenges that Evan probably is trying to mention is this is something we have to find a way to account, but we still don't know how to account, but we have the chance to work on that. But noise is not, it's not something we can eliminate. We are talking about a consortium that invests a lot of money in data standardization and harmonization, and we still have problems. Can you imagine if you don't have any of that? So it's so that's the problem. So the other thing about the pathogenesis versus, yes, that's not a problem that I'm trying to resolve. I think it's important. I just mentioned that. We don't understand disease. I agree with you, right? We have several exquisite investigators that are trying to understand disease. I think the disease is just too complicated. And if we look around, all the diseases are. And I think what's happening right now is a change in the way that we look at pathogenesis and the way we look at data. And I think this new, this evolution and constant evolution and the way that we see even old data now in a different view, it's helping the field move forward in a pace that we haven't had in the last 50 years. So I think we will get to a stage to understand better pathophysiology of TB in a way that we never had before, but it's because technology and also the way that we look at the data will allow us to discover things like, is gamma really important or not, right? And I think we will get to that stage, but we are just not at that stage because we are just discovering how this new technology can help us to understand TB. That's what I tried to say.
Thanks.
Thank you.
Thank you.
Yeah. Thank you very much, Bruno. Great. The data acquisition, is that not a problem for you? Where I come from, South Africa, very fragmented healthcare service, private sector, and then the public sector and different provinces, and nobody wants to share data with anybody else. Then there are ethical concerns. Then there's data quality concerns. And then going beyond countries, there are regions and countries who refuse export data, try to get European data. You have to actually sign on the grant agreement that you will not use any of the certainly personal identifier or anything that can get close to a personal identifier. So those are all challenges that I find difficult to overcome. I'm not sure what it's like in Brazil. Maybe after the next international meeting, I'll know. But how do you deal with those issues?
That's such an important point, Gerhard. So you have an idea. In Brazil, TB care is public, right? So you can go to a clinic, a private clinic, but you will need to be in the national registry to get access to medication. Otherwise, and medication is dispensed in public sector. So I think the government centralized the procedures and information in a way to facilitate that. Highly inspired by the HIV program that Brazil has done. So I think that is something very unique to that region, because we have cohorts in Peru that are quite noisy, collaborative cohorts, because of the lack of this centralized data management. So the way that I found to get access to more, let's say, sensitive information was to prove to the government that I could tell them what to do in a precision that they did not have, right? So I'd say, well, look, I digest the data for you in all the instances that you need, but it's yours. All I ask, if once we de-identify and a concept sheet is approved by the government, that we use that for research purposes. But the main point was, let's work as a contractor for the government to help them in the questions that they want without thinking about us. So it's a fitness expense. Like we spend money, time trying to do that, but at the same time, we gain access to data that we actually never imagined that we would have. All right. So now, if the person is into any kind of program, of government program, we can track that down. If it's receiving a medication for any other disease, so we can do that, and we add cost, and we add absolutely everything we could with that. So that is something that Brazil brought us advantage, and I discovered with time the venues that we could use and how we could do that in a way to please the government, but also to be able to answer important questions that we thought that were important. How to do that at global level, it's still right. So maybe I know South Africa, and I know how complicated it is because of this mixed ways. India is not that simple as well. It's extremely complicated. And I think maybe bringing the Brazilian government and telling to other, what I thought about this meeting in Brazil is, if we can take the policymakers, the stakeholders from the other countries and put them together with the Brazilian authorities for a meeting between these guys, just to exchange how, success and lessons learned could be that platform, because we communicate with all these with these governments. So I think we could do that, but I don't know how to solve that. This is a pretty complicated, but I agree with you. For me, it's the major. If we can share data, if we can have a clear idea of what's going on in a country, how can we design strategies? It's just yeah.
One last question.
I really like, well, firstly, thank you. Very interesting. Thank you for visiting Rutgers. I like the concept of multi-omics integration. I don't totally understand it, but I really like the concept. I was wondering, you showed data from mice and non-human primates and clinical human data. And how does the experimental system, how do they fit into the models that you use? Is a mouse and that's it? Or do you learn something from the mouse that you then can apply to non-human primates and humans?
No, I wanted to show to you that we could use tools that we use in the clinical setting. We could use that in experimental models. That was the point that I wanted to make. But the models are dramatically different. Timelines of the experiments, type of tissue that is committed, type of necrosis, pathways that are induced, a lack of cell types in the lesions. So we never tested intermodal associations. In Eduardo's papers, in the recent papers in Nature Microbiology, we've done the analysis in mice, we've done the analysis in non-human primates, and then we show commonalities, but we never actually put this all in one single package. That would require more communication between the silos to come up with a strategy and how to really compare the models, right? I think the non-human primate investigators are surfing in a wave that is quite interesting. You run experiments in 2 monkeys and you got a Nature paper out of that. So it's to infer disease susceptibility for me is dangerous, right? But that's what it's going on right now. Every granuloma is a data point, right? If we are talking about tissue, and the question is about tissue and what's going on in the tissue, I'm fine with that. The problem is when you want to translate this knowledge to infer disease phenotypes. That's what is dangerous. But I don't know, Jerry. This is something that a place like Rutgers has the potential to answer, right? Because you have heavy and strong experimental groups, right, that performs in vitro and also heavy clinical research going on.
We tried.
Thank you so much.
Thank you. Thank you. Can I stop sharing here?